Um, so finally, I'd like to welcome Anna Peschel from the University of Minnesota with a talk titled Seed Sourcing and Climate Change. What does the evidence say?
Okay, great, thanks. I'll just share my screen here. Okay, one moment. Okay, does everyone see my screen?
Yep.
Okay, great. Okay, well, thank you for the introduction, and thank you all for attending my talk. I am going to present research aimed at providing insight into native seed sourcing strategies for restoration, while also considering how seed sourcing may be influenced by climate change. So, seeds are often intentionally moved between populations for restoration, and this is especially so in fragmented ecosystems where there are few remaining habitat patches that are distant from one another. And this is the case for the remaining tallgrass prairie in Minnesota, which is shown here in red. So when seed sources are remote from the restoration site, we need to understand how plant population fitness changes with distance in order to make good seed sourcing decisions. There are several reviews that have documented the prevalence of local adaptation, but they also make clear it's not always observed. And what we know less about is the magnitude of change in population fitness in relation to the distance it's grown away from its site of origin. So this figure shows different hypothetical scenarios of how population fitness may change with distance. So if there's strong local adaptation, represented here by this dark green line, population fitness would decline rather steeply as it's reared in increasingly distant sites, versus no local adaptation, shown by this yellow line, where there's no change in population fitness with distance. Whereas with maladaptation, this brown line here, population fitness increases with increasing distance away from its home site. So if climate is this main agent of selection, as the climate changes, we would expect to see more maladaptation of populations to their local environment. Some studies have shown that for some species, local adaptation is already disrupted by climate change. And others have shown that sourcing seeds from warmer sites can bolster population persistence. And there are some observational studies that have shown that species are shifting their ranges poleward and to higher elevations, presumably tracking changes in temperature. So while sourcing seeds from warmer sites makes intuitive sense, and there is some support for it, it is an assumption that bears investigating, because what we really do not know for most plant species is how plant population fitness changes with distance. And we want to be making seed sourcing decisions that facilitate the persistence of native plant communities in our restorations, especially as the climate is changing. So I'm going to show results from two studies. The first one evaluates this question of the relationship between plant population fitness and the distance it's grown away from its site of origin. And the second study investigates this question of whether or not plant populations have the capacity in the wild to adapt in their home sites to environmental change. In our recent work with the Minnesota Healthy Prairies Project, we used perennial prairie forbs shown here, which are all commonly used in prairie restorations in Minnesota. And we did a common garden study where we compared source population fitness from different regions within species. We collected seeds from each species from across the prairie in Minnesota. And on this map here, the source population locations are shown color coordinated by region. And we reared a generation in common conditions to diminish maternal effects. And then we planted these source populations into common gardens, where we tracked survival and fecundity of individual plants over 5 years. So I'm going to talk about our results from the Northern Common Garden, which is shown here by this gray dot on the map, as those results are more germane to this question of moving plant populations north as a strategy to promote adaptation to climate change. So we estimated the relationship between source population fitness and the difference in latitudinal distance between the source population's home site and the common garden. And evaluating the difference in latitude captures the difference in distance, considering if the population is sourced north or south of the common garden. And in this example here, the difference in latitude between the source population and the common garden would be negative, as southern latitudes are lower than northern latitudes. So at this northern common garden, we see evidence of climate change as, according to records from the Minnesota Department of Natural Resources, the trend has been an increase in about 3.4 degrees Fahrenheit in average annual temperature per decade since around 1950. If climate is the main factor to which these populations are adapted, we would expect to find a relationship between population fitness and distance at the northern site that looks like this black line here in the figure, where populations from more southerly and warmer latitudes have greater fitness than those that are closer to the common garden. So the question is, given that climate change is happening, do we see that southern populations do better than northern populations at this northern common garden? So now I'll show a series of figures for each species, and I'll kind of orient you to the figure here. So estimates of source population fitness are the points color-coded by region, and figures with stars show where we detected a significant relationship. And the dashed vertical line is the location of the common garden in relation to the source populations. So those with a negative latitude on the x-axis represent source populations moved north. And we found that the relationship between latitudinal distance and source population fitness varied between species. So first, for Monarda fistulosa, shown here, the common name is bee balm. The fitted relationship suggests that populations sourced from more southerly latitudes have greater fitness at this common garden than those closer to the site. Kind of said another way, we see a decline in fitness as distance decreases. For Dahlia purpurea, or purple prairie clover, we found this quadratic relationship, and this suggests that fitness increased as populations were sourced from more southerly latitudes up until a point, and then fitness declined. But we did not detect this latitudinal distance to be a significant predictor of fitness for Dahlia candida, which is white prairie clover. So latitude of the source population was a poor predictor of source population fitness at this northern common garden. So really importantly, while we detected that there were some significant relationships between population fitness and distance, we found many exceptions. You can see immense scatter of source population fitness around the fitted relationships and really large overlaps between some population fitness estimates from different regions. And this suggests that using distance alone to predict plant population success in new areas may be unreliable, and it's not applicable to all species. And this challenges the practice of sourcing seeds based on predicted climate matches as a general approach to restoration. There's also this question of whether wild plant populations have the capacity to adapt to environmental change in their home site without the addition of non-local propagules. And to address this question, we can estimate a population's additive genetic variance for fitness, which is predictive of the change in mean fitness between subsequent generations due to adaptive evolution. So that's represented by delta W on this figure. And that's because it equals the ratio of the population's additive genetic variance for fitness to its mean fitness. So for a quantitative trait like fitness, additive genetic variance is genetically heritable variation, and it's available as a basis for response to selection. Using the annual prairie legume Chamaecrista fasciculata, whose common name is partridge pea, we have found additive genetic variants for fitness to be abundant in different cases. So for the same population when planted in different sites in multiple years, and to manipulated differences in precipitation. And these studies are really important because they show that populations in the wild can possess additive genetic variants for fitness, which is contrary to the historic argument that we shouldn't expect it, as natural selection would wear away any heritable variation in fitness over time. And we now have work that has detected additive genetic variants for fitness in novel environments, more like those we would expect with environmental change. So in this study, we planted one population of Chamaecrista fasciculata into sites along a climate gradient, shown by the dots on this map here. The home site is in green, and the environment is getting warmer westward. Shown here on this figure is mean fitness of this same population planted into each site. And while fitness decreased westward, we estimated significant additive genetic variants for fitness at each site. And this suggests that the populations possessed the capacity to adapt, to increase its mean fitness due to adaptive evolution in future climates without the addition of foreign propagules. So really importantly, when we detect additive genetic variants for fitness, it implies that genetic selection has occurred and that populations have the capacity to adapt to the environment in which they exist. So the assessment of adaptive capacity is immediate to the current environment. So it's still an open question if, given the rapidity of climate change, if the additive genetic variants for fitness we detect is sufficient for adaptation to future conditions. While we only have a few studies that have evaluated the capacity for adaptive evolution, there are other examples of adaptation over short timescales to environmental change, and studies of long-term response to selection that have shown genetic variation persists and that it's not easily depleted. So we should not rule out the presence of additive genetic variants for fitness and the capacity for adaptive evolution in wild populations. This work was put together with my co-authors and with funding support from the Environment and Natural Resources Trust Fund of Minnesota, and I thank you for listening.
Great. Thank you so much for your presentation. So I'll be going through questions that I find in the chat. If people have more questions for Anna, Jill, or Holly, please put them in the chat. So let me see. So I have one from Tonya Brito-Bersi for Jill. So they're asking, I'm wondering what changes do you think the new presidency will bring to this program? I actually think I have another similar question for both Jill and Holly.
Yeah, I would say I think it's still very new and uncertain. I would also point to the fact that our congressional landscape has changed as well. So when you're a federal employee, you help implement the priorities of the executive branch, but also Congress has oversight and funding. So of course our distributed power here in the United States. So actually it's more than just the president changing. So I really can't speculate, but I will say that the work on the national seed strategy, the spirit of collaboration across federal agencies with our partners and others has been going on for years. So I would expect that it would continue in some form, but what that would look like, I really can't say specifically.
That's fair, yeah. And Holly, do you have anything to add regarding your work and the change?
Very similar to Jill's statement of, we're still dealing with the challenges of climate change and seeing changes in fire, flooding, and how to manage the land. So I imagine the questions will still be there and folks will still need our assistance. It'll just depend on what direction or funding is available to do that. And that's all determined by Congress and the president.
Yeah, those are the bigger parts. So I have one for Anna. Does additive fitness need to be evaluated on a per species basis? And could citizen science work and help with this effort?
Yeah, we think about additive genetic variants for fitness for species populations. And we would love for there to be more studies across different species and different environments that address it. It is rather time consuming, as part of what I didn't mention here in the methods is you have to create category populations. So you have to know the relationship among the individuals in your population in order to calculate additive genetic variants for fitness. But I think kind of one thing that we are encouraged about by this work is we now have cases which show that it is there in wild populations, suggesting the capacity for adaptive evolution through genetic change and mean fitness between populations. And it's sort of more encouraging, I think, for us not to neglect that aspect of conservation of populations in the wild as the climate is changing. I hope that answered your question.
I hope it answered Tom Green's question. So all the other questions have been added. Oh no, there's a new one. So Anna, again, for the common garden with the 3 species documenting some adaptation time lags, what were the conditions like in the years you did the experiment? Did they better match the historic conditions? Favored sites? Oh, Anna, you were muted.
Okay, so sorry. So did the conditions in the years of the study match the historic sites of the garden? Was that the question? Yeah, so we also evaluated difference in kind of climate variables, so temperature and precipitation. And so we took the average annual mean temperature during the years of the study that the plants were grown, and we contrasted that to the historic 30-year average of the common garden, both for temperature and precipitation. And we did see that for the northern common garden, conditions were slightly warmer than they were kind of according to historic averages, but the fitted relationships that we found, I answered this in the chat too, but they pretty much matched what we found with latitude, as they were highly correlated. So I chose just to show the latitudinal results, as people mostly think about distance when they are restoring plant communities.
Okay, great. I don't see any other questions that haven't been answered in the chat. So if people have more questions, feel free. Otherwise, we're 15 minutes ahead of schedule.